So let's look at the process of transcription. So in this image, we see this blue ribboned structure. This is our double-stranded DNA molecule. And remember what I said, one chromosome is made up of one double-stranded DNA molecule, but on that one double-stranded DNA molecule, we can have multiple genes. So let's just say that here is a gene region, and we don't need to make a protein from this, so we're not going to transcribe this section. Here is a gene region, and we want to make a protein from this, so we want to transcribe this gene section. The enzyme, the primary enzyme that is involved with the transcription of DNA is called RNA polymerase. And the RNA polymerase enzyme will bind to the region of the DNA that is called a promoter region. And what this does is it in essence kind of lines up that enzyme to be ready to read the specific gene that is going to be transcribed. Since the RNA is single-stranded and the DNA is double-stranded, our RNA polymerase is only going to read one double-stranded segment of the gene region, and when it reads it, it will enable a complementary base to the DNA that's being read to be assembled, forming the RNA. So as we continue with transcription, here we can see the the RNA polymerase enzyme has attached at the promoter region, and it's already has moved down the DNA strand. So our RNA polymerase here was all the way back here, let's say, when it started transcribing our gene segments. So you can see from that RNA polymerase, we already have this copper-colored ribbon that's been formed. And this copper-colored ribbon is our RNA molecule that is complementary to this section, or our gene section, of our DNA molecule. So in this section of our DNA molecule, remember, we'll have a deoxyribose sugar, phosphate, and DNA nitrogenous bases, whereas in this RNA backbone, we'll have ribose sugar alternating with phosphate, and then our RNA nitrogenous bases are going to be the ones that are complementary to our DNA nitrogenous bases. So, for example, if this was an adenine in our DNA, this would be a uracil in our RNA. And if this was a cytosine in our DNA, this would be our guanine in our uracil. So the RNA polymerase enzyme moves down the DNA, unwinding it as it goes, enabling RNA nucleotides to be assembled complementary to the DNA strand that is being read. The area where this is occurring is called a transcription bubble, because it's basically a rounded bubble-like structure where transcription is occurring. The final step in summarizing transcription is, as your RNA polymerase enzyme continues to move down the DNA gene that it is transcribing, it assembles the complementary nitrogenous bases until it gets, in essence, to the end of that DNA gene sequence. When it gets to the end of the DNA gene sequence, it will have finished its task, laying down complementary RNA nucleotides to the DNA nucleotides. So if we just kind of focus in on one area here, we have a nucleotide adenine, and the complementary nucleotide that was laid down by the RNA polymerase enzyme would be a nucleotide that contains the nitrogenous base uracil. And then, of course, to this DNA nucleotide that contains the nitrogenous base thymine, we lay down an adenine, and so forth, until we get to a region of the gene where our RNA polymerase molecule comes off of the gene segment of the DNA and our transcript is released. The transcript, which again we can see as this copper colored ribbon, will leave the nucleus and go into the cytoplasm where it can be utilized. So there's three basic types of RNA that are made through the transcription process. rRNA, which is ribosomal RNA, which helps to make the ribosome, and it helps make ribosome with protein. And recall the ribosome is going to be the structure that actually reads the messenger RNA. So the second type of RNA that is produced through transcription is called messenger RNA. And it is the RNA that most people are most familiar with because it is actually carrying the gene information. It is what is going to be read by the ribosome. The third type of RNA is called tRNA or transfer RNA. And transfer RNA is going to have a clover leaf type shape, and its job is to transfer amino acids from the cytoplasm of the cell to the site of protein synthesis, which is a ribosome. Now on this slide, what we're seeing is when RNA is made, and specifically messenger RNA, when it's made in a eukaryotic cell such as a human cell, before it can be translated, it has to be what's called processed, meaning it kind of has to be finished. So in a gene segment, which remember is a section or a segment on double-stranded DNA, which is packaged inside of a chromosome, that gene segment contains extra information that basically won't turn into a protein when a messenger RNA is translated. So we have to get rid of it. So from the gene, transcription occurs, and we make RNA transcript. And that RNA transcript, as we can see from this image, contains complementary information to our DNA gene. And that complementary information is organized into big chunks or sections that are called exons and introns. The exons are the part that will make sense to the ribosome. So they are going to be kept. The introns are areas that aren't going to make sense to the ribosome, so they are going to be removed in RNA processing. So we can see moving from the transcript to the finished RNA, we have removed the introns and put together the exons. So our messenger RNA transcript has been shortened, but it makes sense now to the ribosome that's going to read it. Notice we also have the addition of what's called a 5' cap and a 3' poly A tail. And what these do is help protect and prepare the messenger RNA to be able to be read by the ribosome. So you might ask, well, what's the point of the introns to begin with if we're going to get rid of them? In essence, the purpose of introns, they believe, is to help reorder exons as well as to assist in a process called crossing over, which we'll talk about in week 5.